right, small intestines in general. The small intestines are about 1 to 1.5 inches in diameter and about 20 feet long. They are found in all four quadrants of the abdominal cavity, if you remember your picture from the first lecture. The small intestine contains many structures that are there to increase the surface area for secretion, digestion, and absorption. These structures include circular folds, villi, and microvilli. First, circular folds, also called plicae circularis. They are formed by the mucosa and submucosa. They are around 1 centimeter tall, so they're visible to the naked eye. They increase the surface area and also slow the passage of the contents of the lumen. They remain even after distension of the wall of the small intestine, so they're a little different than gastric rugae. Intestinal villi are small fingers that cover the small intestinal lining. They are formed by the mucosa and are around 1 millimeter in length. Okay, so we went from a 1 centimeter circular fold, and then each of the circular folds are covered by villi that are 1 millimeter in length. Oh, and here's a nice picture. It's very, magnified because it's difficult to see the villi with just the naked eye when the scope picture is not magnified. So here they really blew up a picture from a enteroscope, and you're seeing these little tiny fingers. Those are the villi. Okay, simple columnar epithelium covers each villus. The cells of the epithelium are called enterocytes. Goblet cells that secrete mucus are located between the enterocytes. Microvilli cover the apical surface of each enterocyte. The microvilli are about 1 micrometer or 1 millionth of a meter in length. So whereas a circular fold was a centimeter and a villus was a millimeter, now we're talking about something that is a millionth of a meter or a thousandth of a millimeter. So these are really tiny. They're on the surface of each enterocyte. In the core of the villus is the lamina propria, composed of loose areolar connective tissue. The lamina propria contains capillaries that absorb nutrients. I mean, what are we here for, right? A lacteal, which is a lymphatic vessel, absorbs substances that are too big to enter the capillaries. Enterocytes combine lipids with water-soluble proteins to form little structures called chylomicrons. I have a picture of a chylomicron in the corner on the right lower corner over here. Lacteals absorb the chylomicrons and carry them through the lymph fluid into the blood. The lipids in the chylomicrons are used by the tissues of the body as needed as the chylomicrons circulate through the blood. At the bases of the villi, in the lamina propria, are intestinal glands or intestinal crypts. The intestinal glands contain cells that secrete hormones involved with digestion. The glands also contain stem cells that continually replace enterocytes and immune cells that protect the intestines. Small intestine functions are as for the whole digestive tract, digestion and absorption of nutrients. 90% of absorption in the digestive system occurs in the small intestine. Only around 10% occurs in the proximal large intestine. And as we already learned, it's not common that the stomach does any either. So really the small intestine's the main site of absorption. The small intestine also protects the body from ingested pathogens and toxins. So while it's absorbing all of these nutrients, it has to be careful to protect the body from ingested pathogens and toxins. Control of the small intestine functions. The secretory activity and motility of the small intestines are controlled by the autonomic nervous system, hormones, and local reflexes. Parasympathetic stimulation via the vagus nerve, cranial nerve 10, increases secretory activity, peristalsis, and segmentation. Sympathetic stimulation decreases secretory activity and digestive movement. Hormones produced in the duodenum coordinate secretory activity in the stomach, duodenum, liver, and pancreas. That will all make sense when we talk about the central location of the duodenum. It's attached to the stomach and also through ducts to the liver and the pancreas. The blood supply to the small intestine is mostly from the superior mesenteric artery. So there's the abdominal aorta, and the aorta is the major vessel that runs from the heart through the thoracic cavity and the abdominal cavity. So there's the abdominal aorta, and then it has many small branches in the peritoneal cavity, and the superior mesenteric artery is found right in the midline, and it supplies most of the small intestine. You'll see on models. Okay, the digestive system. Now let's go through the duodenum, jejunum, and ileum, the three parts of the small intestine. And here they have nicely colored them for you. The duodenum, which is the shortest part, it's only about a foot long. And then the jejunum in purple here, and the ileum in orange here. And they are both a bit longer. You can remember, if it's easy for you, that the duodenum is about 10 inches and that the jejunum is about 10 feet and the ileum is about 10 feet long. It's not exact, but it's an easy way to remember it. So the duodenum, it's the first part of the small intestine. It's around 10 inches long. It's connected to the stomach, and it's also connected to the pancreas and liver through ducts. It's the mixing bowl for chyme from the stomach, digestive enzymes and bicarbonate from the pancreas, and bile from the liver. So they all go into the duodenum, and then they're, churned around, CHURN, around in the duodenum, and they're all mixed together. The duodenum contains duodenal submucosal glands that produce large amounts of mucus. The mucus protects the epithelium from the acidic chyme that's entering from the stomach. The mucus contains bicarbonate ions that buffer the acid in the chyme. So the pH of the acid in the stomach is about 2, whereas our blood is about 7. So, many orders of magnitude more acidic in the stomach than in the blood, certainly. And the duodenum wants to get that pH up a little bit, and make sure that the small intestine that has to do the job of digestion and absorption can contact the nutrients in the chyme without being burnt by the acid in the chyme. Halfway along the duodenum, the duodenal ampulla and papilla are found. The duodenal ampulla is formed by the joining of the pancreatic duct from the pancreas and the bile duct from the liver. The duodenal papilla is the bump over the ampulla. The pancreatic duct releases digestive enzymes and sodium bicarbonate ions into the duodenum. Specific concentrations of acid are needed for pancreatic digestive enzymes to function. Okay, so we have very acidic chyme, but the bicarbonate's going to bring up that pH, and along the way, we're going to hit the right pH for pancreatic digestive enzymes to function. The bile duct releases bile into the duodenum. The sphincter of ampulla controls the release of the secretions from the bile duct and pancreatic duct into the duodenum. The jejunum is the second part of the small intestine. It is around 8 feet long, but you can remember 10 if you want. It has long, closely packed villi, and it performs most digestion and absorption of nutrients. And you see here, I have a little cross-sectional view showing the jejunum, and you can see that the villi are quite long and closely packed. You can compare them to the villi in the duodenum, which are shorter and more widely spaced, and in the ileum, where the duodenum are also shorter and more widely spaced. The ileum is the third part of the small intestine. It's around 12 feet long, but you can remember 10 if it's easier, and it performs some digestion and absorption. Its distal end contains aggregated lymphoid nodules that protect it from the many bacteria that are found in the large intestine. Now, by aggregated, they mean like clumped together. Like an aggregation is like a clumping together. And now here we have this very helpful cross section comparing the duodenum, jejunum, and ileum, and this will be very helpful when you're looking at your histology slides that you have to learn. The duodenum has duodenal submucosal glands, shown here in purple. The jejunum has long, closely packed villi, and the ileum has aggregated lymphoid nodules underneath the mucosa. So that will help you with your histology slides. Okay, moving on to the large intestine. The large intestine is around 5 feet long. Its diameter is around 3 inches. It's 3 times as large across as the small intestine. The large intestine has 3 parts, the cecum, colon, and rectum. Okay, so the large intestine has 3 parts, the cecum, the colon, and rectum. The colon has an ascending, transverse, descending, and sigmoid part. The contents of the ileum pass through the ileocecal valve or sphincter and enter the cecum. The ileocecal valve is usually called the ileocecal valve rather than the sphincter. This is in contrast to the pyloric sphincter, which is called the pyloric sphincter and not the valve. So the cecum leads to the ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and anus. The ileum connects to the cecum. The ileocecal valve or sphincter controls the movement of the contents of the ileum into the cecum. And you see there the little ileocecal valve, which looks like a little button there. We have a nice model in class at Miracosta. The appendix is a small tube that's attached to the cecum. It's around 3.5 inches long and around 1/3 of an inch in diameter. The mucus and submucosa of the appendix contain a lot of lymphatic tissue. So it's thought that the appendix has a role in fighting pathogens, all right, an immune function. The lumen of the appendix may contain beneficial organisms that can repopulate the large intestine after infection with a pathogen, which might have, as part of the infection, have damaged or killed off the population of beneficial microorganisms. The ascending colon extends from the cecum to the inferior liver. It turns to the left at the hepatic or right colic flexure. I like to use hepatic because it reminds you of where it is. The transverse colon extends from the hepatic flexure to the splenic or left colic flexure and then turns inferiorly. And I like to use splenic flexure because it reminds you where it is, up by the spleen. The descending colon extends from the splenic flexure to the sigmoid colon. The sigmoid colon starts at the iliac fossa and extends to the rectum. It is posterior to the bladder and in women to the bladder and uterus. The wall of the large intestine is thinner than that of the small intestine. The mucosa has simple columnar epithelium with many goblet cells. However, the large intestine does not have villi, so the histology looks quite different. The mucosa contains deep vertical intestinal glands. The glands contain many goblet cells that secrete a lot of mucus to lubricate the passage of undigested waste. And when you're looking at the histology slides, it can sometimes look a bit like there are villi, but what you're actually looking at is deep vertical intestinal glands. The lamina propria of the mucosa and the submucosa contain large solitary lymphatic nodules that protect the body from pathogens. And you will be able to see this. The muscular layer of the colon has a circular layer and a longitudinal layer. But the longitudinal layer only contains 3 bands of smooth muscle called the taeniae coli are on the outer surface of the colon under the serosa. Taeniae coli aid in peristalsis and segmentation. They maintain strong muscle tone that creates haustra, or little bunches or pouches in the colon wall. Haustra perform segmentation and churn the contents of the lumen. And we have a nice real model of the cecum and ascending colon. And you can see Taeniae coli in Miracosta with that specimen. The rectum is the last part of the large intestine. It is around 6 inches long. The rectum is usually empty. Movement of feces into the rectum triggers an urge to defecate. The abdominal aorta leads to the superior and inferior mesenteric arteries that supply the colon. So we already saw the abdominal aorta and the superior mesenteric artery when we were talking about the blood supply to the small intestine. And now for the blood supply to the large intestine, we see that the abdominal aorta leads to the superior and inferior mesenteric arteries, both of which supply the colon. Large intestine functions. The functions of the large intestine include absorption of water, electrolytes, and vitamins, which are produced by colonic bacteria. In addition, the large intestine stores feces prior to defecation. Movements of the contents of the colon. Waves of peristalsis move the contents of the colon from the cecum to the transverse colon rather slowly. This gives time for water to be absorbed. Stronger waves of peristalsis, called mass movements, are caused by distension of the stomach and duodenum, and mass movements move the contents of the colon from the transverse colon to the rectum. And finally, the anal canal and anus are the last parts of the digestive tract. They are not part of the large intestine, but they're just a bit at the end. They follow the rectum. They are around 1.5 inches long. The anal canal has simple columnar epithelium, whereas the anus has non-keratinized stratified squamous epithelium to help protect it from any rough feces that might be passing through. The anus has a circular internal anal sphincter, and that sphincter is smooth muscle and involuntary, and then a circular external anal sphincter that is composed of skeletal muscle and is voluntary. The defecation reflex. When feces move into the rectum, stretch receptors are stimulated, triggering an urge to defecate. When an urge to defecate is stimulated, the internal anal sphincter opens involuntarily. Feces move into the anus. The external anal sphincter opens under voluntary control, luckily. And that is the end of the lecture.